Hello, today we're going to talk about the recent research conducted by Dr. Christina Hilden at the University of Helsinki about plant biomass degradation by white rot fungi. So why does plant biomass need to be degraded? As we can see from the diagram, plant biomass is an essential part of the global carbon cycle. In order for carbon within plant biomass to re-enter the cycle, it must be degraded by decomposers, in this case, fungi. The problem with plant biomass is that it can be very difficult to break down completely because it contains many complex carbohydrates such as lignin. As we can see, lignin has a very complex organic structure with lots of cross-linked phenolic polymers. This means we can't rely on animals to break it down through digestion, but we have a solution. Fungi have the ability to produce enzymes that are able to degrade plant biomass, including lignin. There are two main groups of fungi that are capable of producing plant-degrading enzymes. These are the ascomycetes and the basidiomycetes. The main enzymes used by these fungi are heme peroxidases, laccases, and cellulases. Together, these enzymes are able to degrade plant biomass, including lignin. To study these fungi, samples were collected by Hamby, the University of Helsinki's Faculty of Agriculture and Forestry Division of Microbiology and Biotechnology. They've managed to build a collection of over 2,000 different isolates of white rot fungi from all over the world, including Brazil, Indonesia, and China. Biochemical reaction tests of the isolates showed different reactions to plant compounds even within the same fungal species. We can see the differences in this photo of the reaction assays, but we don't know why the differences are occurring. As we already know, plant cell walls are made up of many different molecules, like cellulose, hemicellulose, lignin. We want to know how the extracellular enzymes produced by fungi selectively target the organic molecules within dead plant cells throughout the different stages of fungal development so that we can isolate specific enzymes and use them to extract desirable products from plant biomass decay. For example, high-grade lignin has a highly profitable market value when used to make certain things like carbon fiber. We mainly use molecular techniques to analyze the genomes of fungi throughout the process of decay. This allowed us to observe the substrate and time-dependent production of extracellular enzymes. One species was Dicomyces squalens. This fungi produced extracellular enzymes in both liquid and solid media. The genomic and proteomic analysis shows enzyme production was dependent on the substrate and time. We found the genes of the carbohydrate active enzymes were involved with selective degradation of biomass molecules. For example, lignin degradation only occurs during the initial growth phase. Then other biomass molecules are degraded by the fungi. Other species that were studied were Oba rivulosa and Flebia centrifuga. Flebia was able to tolerate colder climates and able to still degrade biomass. And genome analysis of Oba shows that an isoform of carbohydrate active enzymes were present, similar to the forms found in Dicomyces. Final species considered was Agaricus bisporus, which you might be familiar with as button mushrooms. As a secondary decomposer, Agaricus was only able to grow on plants that were already partially decomposed. It's commonly found in leaf litter and requires high nutrient and humidity levels. Genomic analysis of Agaricus found genes for laccases, copper radical oxidases, and manganese peroxidases. These are all involved in plant biomass degradation. In addition to the roles that fungal decay play in the carbon cycle, identifying the genes by which lignin can be selectively degraded by fungal enzymes can allow us to turn off the production of lignin-degrading enzymes and extract lignin from plant biomass to be used as a useful product. Lignin powder can be used in the production of carbon fiber, which can be used in many other manufacturing processes, as well as lignin being used to artificially produce vanillin, which is used as a cheap synthetic flavoring. Hopefully this has opened your eyes to the wide range of fungal biotechnology applications and the importance of plant biomass decay.